Hello everybody. Nice to see you again. Some of you last time we saw in Singapore. I'm going to talk about our project, which focused on gut health and performance of chickens raised on black soldier fly larvae. And this work was, led by my former master's student who graduated this past summer, Cameron Beamsley, with a lot of help from Molly Cobb, another grad student. And we had a lot of assistance from Chris Bailey, who was our avian nutritionist, and of course, Jeff. I'm going to give you the latest on what we've been doing. And, as we are working on writing this up into a publication, hopefully that'll be something we'll share with the community as well. So these are the folks, just to put a name or a face to the name. I'm going to start with kind of briefly talking about why I was very interested in pursuing this particular project with the CEIF, and why I'm kind of passionate about pushing this in the poultry space. As you folks might know, poultry is obviously one of the most common and popular sources of animal protein in the world, especially in the United States, and it requires a huge amount of feed. And some of the things we know about the animal feed industry is that a huge amount of the land that's available or for cultivation is currently used for animal production. And actually the producing of the feed itself is the majority of it, not actually the footprint of animal farms and so on. And there is a very stiff competition for these primary feed ingredients. And insects obviously are a very interesting alternative for plant-based proteins. So soybean and corn, for example, are the two main energy and protein sources in poultry diets, as similar to some extent with the swine industry. And there's an increasing amount of competition for the soybean because there's only so much land and water to keep producing the amount of soybean required. And the other part of this is one of those things which, being able to make insect proteins a feasible option for the poultry industry is very exciting for other reasons, because the size of the poultry industry, the potential environmental impact of being able to make this possible is really large. And that's one of my main interests in doing this. And there's a lot of studies previously on the feasibility studies on using insects as protein sources in poultry industry. But there's still many unanswered questions, and we went into some of these questions, which I'll be talking about today. So here's just a brief snapshot of the meat consumption in kind of landscape over the last 60 years. So you can see that while the consumption of protein sources like beef or pig meat, they haven't appreciated that much. Poultry is actually the biggest consumed source of meat in the world. In fact, the average American consumes over 110 pounds of chicken every year. So that's a large portion. And this is possible by the production of 9 billion plus broilers. So broilers are meat type chicken. So in the poultry industry, there are specialized breeds which produce eggs and specialized breeds that are raised for meat. And broilers are basically what we're looking at here. So those produce for meat. And we see the same trend across the world. Poultry is replacing other sources of meat rapidly, and it is definitely increasing consumption. So if you follow this trend line, one of the big things is that we see in many reports, including by the FAO, is they predict that by 2050 we'll need to produce 50% more calories on 30% less land and water. So this is actually one of the challenges that is motivating for me to figure out alternative solutions for how we can continue to meet the national demand or the global demand for high quality animal protein. Now, the other thing that is very interesting is obviously with CEIF, I don't have to talk too much about food waste. Everybody has heard about the global problem of food waste, but agriculture, because of the impact that it has, it's very crucial for the climate equation as well. So you can see here how emissions from food is one of the big parts of how we can tackle global climate change and reduce greenhouse emissions. So you can see business as usual. You have 1356 billion tons produced, but by implementing one or multiple of these solutions, we are able to, or we're predicted to be able to reduce some of these impacts. So high yields, so in animals or plants, improving the traits, make them produce more eggs or meat on fewer or limited resources, fewer calories. Food waste, obviously, that's not a surprise to anybody. It has a huge impact on greenhouse emissions. Improving the calories, the quality of the calories, improving farm practices, and so on and so forth. So you can see that even adopting partial solutions from this list can make a huge impact. I think I'm interested in is obviously using the opportunities presented here of how we can improve the farm practices while also reducing food waste and what that can do in terms of the poultry industry itself. Again, given how big of the slice of animal protein it is currently. So now I mentioned earlier that insects have been used as animal feed like for over 100 years. I think I've seen some citations from the 1930s of people feeding mealworms to poultry especially, and the benefits that it could have. But of course, black soldier fly is now one of the leading species which is domesticated for this purpose and being produced at much bigger scale than ever before. And again, these are not new things for this crowd, but I just want to emphasize that people have looked at the use or the feasibility of black soldier fly in various animal models, so fish and poultry, pigs and cattle. And we know that there are like no problems replacing plant-based proteins in animal diets with these larvae, black soldier fly especially. A few examples that I can talk about is, the people have specifically looked at the protein and fat content in black soldier fly as supporting healthier growth in pigs and cattle. So performance things that the industry might care for. In the aquaculture space, we have listened to Dale Gatlin's group and their work on the benefits, but beyond that, there's also the growth, performance, and what it means to sustainable alternatives to fishing and conservation of marine resources. So there's data on this. And more specifically, in terms of poultry feed, again, like I mentioned, for over 100 years, people have looked at using insects as protein. I like to always point out that chickens whose ancestors are jungle fowl, in the wild, 67% of their diets are actually comprised of insects. So this is one of those things which makes total sense in terms of they're pre-adapted to utilize insect protein. And then we see that some of these recent studies are also looking at the other benefits of black soldier fly. Black soldier fly supplementation increases the concentration of beneficial short chain fatty acids like butyrate, which are considered to be important or at least indicative of a healthy gut microbiome. And it also seems to be better for gut physiology indicators, such as the higher villi height and crypt depth, so features of the gut epithelium. And these also have benefits for how well these animals perform in terms of being efficient as they're able to absorb nutrition better. So this is kind of the landscape in which we were entering this particular topic as well. And for us, we had a few specific project goals. One of them was to actually look at things are a very different end of the spectrum in terms of inclusion levels. So many of these studies that I referenced, they thought that, or they at least looked at black soldier fly inclusion up to 50% in some cases, but the majority of them between 10 and 25%. Now, of course, I got to credit one of our industrial advisory board mentors who posed the question early when we were developing this project that for the feasibility angle, given how many chickens are raised just in the United States, we would like to know whether any of these benefits are possible at very low levels of inclusion. Because even a 0.5% replacement of plant protein with black soldier fly is going to require several metric tons on the scale of the US production. So this is not something that we are anywhere near producing, but from the angle of what are the performance and the synergistic benefits for gut health or inflammation, this is what got us into this space. So in our study, we looked at the specific inclusion levels, 0.5%, 1%, 5, and 10%. And besides looking at the growth patterns of these broilers raised on these specific diet levels, we also looked at the gut microbiota membership to see how stable or what changes we can observe in terms of the gut microbiome, and also looked at markers of inflammation and stress in these birds, because broilers are raised anywhere from, based on the end market that these broilers are raised for, anywhere from about 35 days to 50 days. So it's a very short production cycle. But in this time, broilers experience a lot of issues. Primary among them, they can have high levels of morbidity, gut health issues, higher levels of inflammation. This is because of, first of all, the amount of feed that they are ingesting on a regular basis, on an hourly basis. They're eating around the clock. And then obviously there's some research suggesting that diets which are high in soybean content also promote inflammation and disorders such as leaky gut condition. So we were interested in contrasting, whether there are any benefits to the amount of inflammation or the stress that we are going to observe. And we did this by a longitudinal sampling approach. So this is also one of the ways we distinguished our study from previous studies. There's a lot of terminal studies which looked at, let's feed these to the birds at the end of the study or at slaughter weight. We're going to assess these things. We differed from that in a few important ways. We actually looked at this in a longitudinal series, especially for the microbiota diversity, to look at patterns of stability, how it changes, because broilers' diets are changed, around the 4th week when they transition from a starter to a grower diet, for example. And we also looked at kind of not as many, points of sampling, but midpoint and an endpoint sampling of the plasma markers for inflammation and stress. So I'm going to call this the trial 1, because later on we'll get back to talking about trial 2. So we had these 5 treatments: control, which was a standard industry diet, which is soybean and corn based, and 4 insect meal inclusion levels, where we basically had the diets formulated such that we were replacing the soybean fraction and adjusting for the fat content of these diets at 0.5, 1, 5, and 10%. Again, we were not sure how different these diets would be in terms of the final proximate qualities of these diets, the fat and protein content. But after we analyzed the diets that were formulated, we actually figured out that all the diets that were within like 2% of what we were expecting them to be in terms of their fat content, because that's one of the things with whole black soldier fly we got to be aware of. And we measured weekly growth biomarker profiles at 2 intervals and microbiota analysis based on weekly sampling. And we looked at a total of 300 broilers that we started the study with, and they were evenly split across these 5 treatments in a randomized block design. So they were in different rooms to avoid cage effects, of course, between treatments, because in any microbiota study, you don't want to have treatments kind of in different rooms. That itself drives one of the major patterns we see. So that was generally our experimental design. So here's some quick pictures of the mixing of the feed at the higher levels of inclusion, the 10%, for example. We had to improvise a little bit to get the feed to be a good consistency and for the pelleting process. Here's like a picture when the chicks were placed and they were marked after we had randomly divided them. And here's another picture showing our experimental pen setup here. And here's, chicks at a week old, slightly older broilers. You can see the blue marking as, sometimes they can, jump over or, be escape artists in these pens. So birds were marked so we could keep the treatments, the birds in each of them apart. And here's Cameron, who was taking a cloacal swab from one of these chicks. So let's get into the results of this particular study. So one of the first things we looked at was obviously we tracked the performance, and what we see is the trajectories differed slightly, but the finishing weights at the end of the trial at 7 weeks were not significantly different. So they were within 150 grams of each other between diets. We also performed analysis of the curve itself to see if there was like significant differences in basically its rival curve here, and we did not. Although there were apparent differences in some diets, they were not statistically different from each other. So this was a good sign in terms of we again confirmed other studies which talked about black soldier fly being as good for growth, if not better in some cases. In our case, we showed that the controlled diets, or our experimental diets were not really different from the controlled diets in the kind of growth that they helped achieve. So this was good in terms of knowing that the birds were all good. We didn't have any morbidity or mortality, unexpected mortality in the study. So all those things that we are interested in, we didn't have any other problems there. Now, looking at the biomarker profiles, we looked at 3 specific markers. So you can see here interleukin 10 and interleukin 12. Now, these are both inflammatory biomarkers. And one of them is a pro-inflammatory biomarker. The other one's an anti-inflammatory biomarker. So interleukin 10 is the anti-inflammatory one. And in these panels, we can see that we have the diets laid out on the x-axis, control 0.5, 1%, 5%, 10% inclusion, and the concentration measured by ELISA. And what we would expect to see in these kinds of data is anti-inflammatory markers should be higher if they are suppressing inflammation. And on the other hand, we should see a reciprocal picture with the pro-inflammatory markers as seen in the IL-12. We saw a very nice relation here in that we see that in the controlled diets, there was no significant differences between the 2 time points that we looked at, but in our different levels of inclusion, we see that there's this relation. We see that the anti-inflammatory markers are higher at the end of the study than in the middle of the study. So this means that we see that black soldier fly was actively or being invoked in suppressing inflammation. And we see an opposite outcome in terms of IL-10 results. And we see that in these cases, these differences between the time points were statistically different as well. And not so for the 10% inclusion for inflammation markers. So I want you to keep this in mind. Now, that's good as far as the inflammation goes. So we see that even at 0.5%, we are seeing, a observable differences in inflammation control. Now, when it came to the corticosterone, so in mammals, this is cortisol. In birds, corticosterone. This is a a stress biomarker. And higher values of corticosterone means that higher levels of stress. And so what we see here again is, contrast to the control, where the levels of stress were much higher in those birds. But in the other inclusion levels, we see varying levels of stress towards the end of the study. And in this case, the 5% and the 10% inclusion diets had the most beneficial profile in terms of regulating corticosterone, significantly so. And now, obviously, this is suggesting that the inclusion levels have a differential effect on how they're regulating inflammation versus stress. So how do we decide what is a optimal level of inclusion? So this is something that we looked at. We used a decision tree approach and a couple of other approaches as well to determine what the best inclusion level is based on the biomarker profiles. And we thought that if you are a producer who's finally interested in the morbidity levels in the broilers, maybe you're going to think inflammation is more important to control. So we weighted the inflammation markers at 40% each and the corticosterone at 20% in this particular analysis, which helped us generate a ranked list of optimal inclusion levels. Now, of course, you should notice that in this case, we intentionally said inflammation is more important. So we see that with this 5%, 10%, and 1% were seen as the most optimal diets in that order. Now, we also did this approach without providing any preferences. I have presented this previously at one of the CEIF meetings. If we didn't provide weights for these, we just went in with this. It basically swapped around this order very slightly. 10% was seen as the best one because it seems to have such an outsized effect on the corticosterone levels. Now, in a different approach, we also used a ridge regression approach, which also supported a 5% inclusion as the optimal inclusion level. So it had the best profile overall based on things that we were considering. So now, as far as the gut health aspect, so we went into looking at the cloacal swabs. So we had over 600 swabs total across our sampling design, and we sequenced the V4 region of the 16S rRNA gene, and we had extremely good quality data. So here you see a histogram showing the sequencing depth, so averaging 60,000 reads per sample. This is more than twice what most 16S microbiota studies use, and we had very good quality data. We are very concerned, especially because in our previous studies, we know that cloacal swabs can be a little tricky because they are not especially representative of the lower GI tract, but we wanted to be able to make better inferences, so we went for this high sequencing depth strategy here. So we'll recover as many taxa as possible. And with this approach, we did recover a total of about 10,000 plus OTUs, which were altogether classified into about 1,100 taxa in this case. And when we looked at the longitudinal series using diversity indices, so which is presented here. So I'm presenting Shannon diversity, which is a commonly used measure of microbial diversity. Now, Shannon diversity it looks at both the richness and the evenness of those taxa within samples. And we can kind of look at the trends here to see what's being picked up and whether they're similar or not. So what we see interestingly is that in the controlled diets, we see that it starts off a little lower, but it kind of gradually goes towards an increasing number of taxa. And this was very interesting because we do hear in the literature that higher diversity is better. And whereas if you contrast that with the other inclusion level, so 5%, it starts off at a slightly higher level and it remains more or less stable over time. We see the similar pattern with the 0.5% and also with the 10%. And whereas the 1% was also slightly lower to start off with. Now, this is interesting. Again, this is a kind of a big picture. What does it mean? Well, that's interesting to contemplate. One of the things that we see, and we'll talk about, is obviously that in the controlled diets, it starts off low, perhaps because of the microbial population that are successfully colonizing are quite different. And we have seen some data in our secondary analysis that this, in the very early weeks, there seems to be some differential enrichment of taxa between control versus all the other black soldier fly inclusive diets. Whereas there are some taxa which you can consider as disruptive to a normal gut functioning, which increase in the controlled diets later on in life, which doesn't appear to be the case in the BSF inclusive diets. Looking at what kinds of taxa there were and big picture associations in this data, we see that, for example, lactobacillus, which is commonly considered to be a beneficial or probiotic strain of bacterium, was kind of showing a inverse relation with some other microbes here. So, for example, Clostridiaceae. Now, there are members of Clostridia which are commensal in chickens, and there are also members of Clostridia which can be pathogenic. Our data was not able to identify down to the species level what they were, but this is at the family level. We see that these are there are some inverse relationships which are interesting. Again, this is not by treatment, this is by overall data set. Now, when we do look at the treatment level differences, we see that actually 10% appears in this hierarchically clustered dendrogram. 10% is very distinct from all of them. So if you aren't used to looking at dendrograms, you can flip this particular tip in either direction. It doesn't really matter. What we do see is that the branching pattern is indicative. So 0.5% and 1% were most similar to each other, followed by them being similar to the 5% diets, and then followed by control. And therefore, the 10% seems to be quite distinct, between these treatments. And we also see some patterns right away with some differential enrichment patterns. So, for example, in the 1% diet, we see that lactobacillus was in fact enriched, whereas the other treatments, especially the 10% treatment, wasn't that great on this specific taxon, this family. So we dug into this type of differences between treatments a little bit more. Again, this is based on the entire data set. I couldn't break it down by week in this talk because I have more things to talk about. But when we looked at the overall data set, we wanted to see what these differentially enriched taxa were. And one of the things we saw is that we got these 3 taxa at the genus level, which is Varibacillum. I hope I'm reading that right. Yes. And Rubinococcus, and this one, I think I can't read. Pardon my eyes, but all of these were differentially enriched in the 10% diet. So the effect of these enrichments are basically looking for log-fold differences in specific taxa when we compare in a pairwise fashion which taxa appear to be very much more prevalent and abundant in these treatments compared to the others. So in this case, 10% diets were the only one that showed differential enrichment of taxa, and these were the 3 main ones. Now, Varibacillum is not a very well understood taxa, but being a bacillus, there appears to be some similarity to other bacilli that have been described, and I'm not entirely sure what to make of this at this point, but some literature that I came across suggests that it is potentially a beneficial one in terms of promoting some short chain acid and improving gut homeostasis. But again, that is something that those types of validation studies are different. We can only infer based on the abundance pattern here. Now, we also looked at a different approach using a machine learning approach across all the time points, across all the treatments to see if specific taxa were indicative of any of these diets. And one of these taxa came up in this type of approach, and it's called Oceanobacillus. Now, Oceanobacillus is something that's previously been described in from marine environments, some extreme environments. In this case, why are we seeing this? I'm not entirely sure. A couple of things that we have tried to explain with this is one, that the inclusion of black soldier fly is definitely a new challenge for the gut environment, and the challenge is being met by the promotion of specific taxa which are able to colonize when there's an abundance of insect proteins, and Oceanobacillus may be suited to the pH levels that are found in the gut environment. This is one explanation. The second explanation, looking at where these things are found very abundantly, they're in the 5% and the 10%, they seem to be very, very obvious, is that they are correlated with what's found in the black soldier fly larvae itself. And that opens up a very interesting idea that this approach can help us trace back specific taxa to the origin of where the black soldier fly came from. So, anyway, this approach yielded some interesting differences in the specific taxa. So some take-home messages from this trial: we've found that the lowest inclusion level did show some benefits for controlling inflammation, but 1%, 5%, 10% were better for both inflammation and stress markers. 5% and 10% diets suggest microbial community adaptation and suppression of pathogenic microbes. And these inclusion levels that we tested are much lower than what was previously suggested. And therefore, we think that in considering the benefits for inflammation and quart concentrations, that these lower concentrations are actually very promising as an avenue for inclusion in poultry diets. Now I'm going to move on and quickly move through trial 2. And based on the approach or the findings from the first trial, we decided to go with 5% and 1% inclusions and we looked at an additional variable, which was the effect of storage of the larvae. So in this case, we performed a similar study, but we only compared control versus 5% and 1% levels, but we also compared a batch of black soldier fly larvae that had been stored for 1 year versus something that was procured, right before the study. Now, I do not know how long the BSF producers are storing them before they ship them. That's a question I would love to know the answer to, but they were stored for 1 year in our lab before we use this. And again, we looked at weekly growth, biomarker profiles in a longitudinal fashion, and similar study design with 300 broilers split evenly. So I want to quickly run through these results because I have a couple more interesting things to talk about. So in this case, we also looked at Shannon and chow diversity. The chow diversity differs from the Shannon diversity in that it is not looking so much for evenness, but it does prioritize the existence of rarer taxa, those that are very infrequent in the samples. So what we can see here is a very nice, time series as the birds get older over time. So weeks here corresponding to the weeks of the year and the median diversity. And you can see blue is one of these, which is the controlled diet, and the other ones are the other diets. We can see that there is a nice progression of the microbiota over time, increasing in this chow diversity. Now we also see that initially the controlled diets were, in this particular trial, They were at the same level or slightly higher than what we saw in the other diets. This is the, I guess, normal variability we expect to see in birds, and especially with cloacal swabs. And as time goes on, we see a lot of these fluctuations. Now, this pattern is super interesting for anyone who's interested in microbiota studies, because you could be looking at differences at this point between treatments, and you would get the completely different picture. And this kind of emphasizes why we need to consider these types of longitudinal analysis. But anyway, at the end of the trial, we do see that it appears to, I mean, this is the end of life for a broiler, so we should hope that this pattern has stabilized. But we see that, for example, the control and the 5% old larvae showed up the highest diversity in this case. This increase in diversity in the control is similar to what we observed in the first trial. So that's something to keep in mind. It appears that black soldier fly larvae are definitely either suppressing the number of taxa or maintaining a certain number of taxa at stable levels, whereas the control diets seem to keep increasing the number of taxa. And when we looked at the hierarchical clustering, again, a very different and interesting pattern. So you can see here, these two new BSF batches were completely distinct from the control and the other ones. So what's interesting is the control diet was actually very similar to the 1% old diet. So this is cut off here, but I can see ONE and VE%. So 1%, 5% control. So control diets were very similar to the 1% old BSF. Now that is in itself a really interesting outcome here, because to me it suggests that whatever's making the 1% new distinct has kind of faded away in the older larvae. So potentially beneficial compounds. I know we talk a lot about lauric acid, for example, or it could be the impact of oxidation of specific, nutritive properties. All of these are potential explanations for this pattern, but anyway, it seems like they become less effective in terms of altering the gut microbiome profile as they get older in terms of storage. And when we look at these in terms of the microbial compositions, also you can see this big picture showing some interesting differences at first view. So 5% new, 5% old, right there, some clear differences. 1% old, 1% new, again, very clear differences in the kinds of taxa that are abundant in each of these groups. So clearly some big differences between these treatment levels. Now, when it came to a similar enrichment analysis, we see that actually in this case, there were some very different patterns of enrichment. The control diet and the 5% old diet were the only ones that showed significant enrichment of any taxa. And in this case, they were of the Campylobacteria, members of the Campylobacter. Now, Campylobacter is a common microbe. It's commensal in chickens. It is not really harmful to the chickens, but it is a common pathogen associated with foodborne illnesses, next to Salmonella and Listeria and E. coli. So from a food safety perspective, you can see that the controlled diets and the ones with the older BSF both showed, again, these are significant enrichment compared to the other diets, whereas the newer BSF inclusion levels didn't really have the same pattern. So they were better at suppressing. Now you can think of these types of key taxa and what impact they have on the microbiome of the chicken. And this is one of the potential explanations for why we see changes in diversity patterns as the birds are getting older in the control diet, because you have certain keystone taxa which basically have a lot of influence on changing the community structure. So in summary, we uncovered a few previously unreported things. We have this very interesting interaction, which I like to think of as synergistic benefits of black soldier fly at low inclusion levels for stress and inflammation, and evidence of probiotic enrichment in the very bacillum that we observed in the 10% diet, but that's something to be validated, verified. We did not pick up the same levels of varibaculum in the second trial, but also because we did not have the 10% diet there. We did see that in the second trial that having newer batches of black soldier fly, even at 1%, seems to suppress Campylobacter and similar taxa in those birds. So that is very interesting angle for the folks who are interested in food safety. And then we saw some other differences between the newer versus stored BSFL on the microbial communities.